Okay, so welcome. Today we have with us Dr. Sergei Pustylnikov. He's co-founder. Sophia Lane is not with us. Oh, actually, I guess she will be joining us. But they are going to talk to us today about their new venture, 100x Bio, 100x more efficient T-cell research, and a little bit about Sergei is he's an immunologist for over 15 years working at academic and industrial laboratories, specializing in T cells and antigen presentation. His startup is striving to generate T cell data on antigen specificity and matching TCRs 100 to 1,000 times more effectively than before. The title of his talk is Autoimmunity Applications of New Efficient Method for Generating the Data on T cell antigen specificity and corresponding TCRs. And we look forward to hearing from him. He has given a private presentation to a couple members of the T1D scientific community whose work focuses on T cell receptors and antigen specificity. And they have reported back with rave reviews regarding what 100x Bio is involved in. So thank you very much, Sergei, for joining us. And I see Sophia also. And we look forward to hearing more about this fledgling company you've got going.
Thank you very much, Monica, for your kind introduction. And thank you for inviting us. Company 100x Bio is indeed trying to very efficiently capture the T-cell antigen specificity data, and downstream, it optionally offers finding the sequences of the corresponding TCRs. Basically, how we make it 100 times more efficient, the concept is very simple. We want to retest the same sample multiple times. But I will get deeper into technology a bit later, but how this is maybe relevant for autoimmunity research. So first of all, this is hypothesis, but if it all works as expected, we can help efficiently find exact immunogenic peptide sequences of autoimmune proteins. How this is helpful downstream is that we can develop the target-specific therapies, for example, tolerizing vaccines or antibodies against those TCRs of the cells, of T cells, autoimmune T cells. And we also can track the disease progression. And just thinking of how it compares to TCR sequencing, imagine in one hand, you can run the sequencing of the TCRs in this blood sample and check the disease progression by running sequencing, or alternatively, you know exact autoimmune peptide sequences. So you take the PBMCs, add the specific peptides, and you see how many cells respond. It's going to be so much cheaper and so much easier to scale. Okay, now moving forward, me and Sophia, we both, most of our lives are academic scientists, and I worked in academic labs about 15 years. And I think my main interest was always how T-cell responses are impacting the disease and how T-cell responses are involved into, let's say, building the new therapies. And for Sophia, I think her main focus, and Sophia, correct me if I'm wrong, is T-cell receptors, this exact event of T-cell activation, and then bioinformatics, sequencing of TCRs, and analyzing the intricacies of T-cell activation. So we are working on this idea of multiplexing T-cell activation. What if we can test in the same sample multiple T-cells activated by multiple different epitopes? So why, in general, people are studying the T-cells? What are they asking? So they want to know how many T-cells react, what are the exact T-cell targets, what's the activation outcome, and what T-cell receptors get triggered. But usually this data is used, of course, in basic research, it's just used to understand how the whole immunity works. But in applied science, the data is used for developing of new therapies or developing of new diagnostics. And this T-cell response data helps to control efficacy of different drugs like vaccines and safety of different other drugs like biologics. Biologics can induce the T-cell responses, which we don't want. Cell and gene therapies can induce immune responses, which we don't want. And all these studies are done in both preclinical and clinical settings. And what is interesting is that those preclinical and clinical projects, they can be very expensive. The normal cost can be $10–100 million. So biosamples come as very expensive with limited volume, and very often they are unique. And if you have a lot of people working in academic science, you also, I guess you have some sort of problem accessing the clinical samples, and they come limited. And what is even more limited is the proportion of T cells in those samples. And what is even more limited is the proportion of T cells responding to one single certain antigen, one single epitope. So usual T cell target studies, when people are using the exact single peptides, they must be super inefficient when we do the sample aliquots, because in each aliquot, it can be just 10 cells responding per million. So we waste most of this sample just normally, and there was no other way to do that. Some people can argue that there is a barcode tetramer staining, but it is not actual real T-cell activation. It's a little bit different technique. So in a normal T-cell activation setting, all those approaches are very ineffective. And this, of course, leads to loss of money, time, and data. And many companies really ignore individual targets. And I know a little bit of the work of Adaptive, and Sofia knows more than me, that they are just trying to make real big data correlations and incur that what they find is true. But to confirm, to validate what they find is true, it's really helpful to know exact epitopes which T cells are recognizing. And so many companies ignore individual targets. They say this is antigenic protein, we work with a whole protein, or we work with antigenic pools of peptides which are covering all the sequence of protein. Or they have to pay this huge inefficiency cost of aliquoting the sample, making very complex experiments with peptide pools or individual peptides. So our solution is, what if we can do the repeated testing of the same sample 100 times? So we have 100 targets, unique limited sample, and we add the peptide number 1, we do the quick readout of data, and then we add the peptide number 2, we do the quick readout of data, and then we add peptide number 3, and what if we repeat it 100 times? And then we found a way how to organize this simple idea into a working prototype. And I will show it in more detail downstream. And we also found that further work with the sample, which would provide us a phenotyping of these reactive T cells and TCR data of these responded T cells, matched to exact targets which activated T cells. And our method, as we see it, should be automated, because 100 cycles, we cannot pipette 100 cycles. It is very simple to understand, and I'll show you. And it seems to be very scalable because the reagents are cheap. It's mostly peptides and compatible with all of the existing T-cell analysis methods. So why no one else did it before? Many people ask me if it's such a big inefficiency. First of all, sample recycling, it's not a straightforward idea. It's normal to aliquot samples to run clean experiments. And this experiment of reuse of a sample can be seen as dirty and unpopular, but we have a strong reason to go with that. Because first of all, we have a huge limitation of this very expensive sample. Second, the interaction of TCRs and targets, they are very specific. There is some cross specificity, but it's not a huge problem. Second, this method the idea came with only automation in mind. We cannot expect real person to do the pipetting. And now it's not too many methods developing for the labs only for automation. Then we found a way to do that quick readout, and quick readout, they are known for ages. So we are proposing, first of all, the calcium flux fluorescent readout. It's known from 90s. But no one is using that because people in T-cell research are focused on cytokine detection because they want to know the type of activation. And cytokine detection, it needs time to develop. We need at least a few hours to have the readable amount of cytokines. So we are using the fluorescence to do the very quick readout of activation to make this cycle short enough to make it repeatable. And then we are also developing the gentle wash, which like the wash is traditionally a centrifugation, but centrifugation leads to losses of the cell, losses of viability, and losses of functionality. So we found a way to remove the previous treatment and add the next treatment without losing any cells. Now, how we exactly do it? Imagine the transfer inserts. Many people worked with them for migration assays. And for migration assays, those inserts are having the membrane, which is permeable for the cells, and the cells are seated at the bottom of a membrane. They have some stimuli to go through the pores and reach the bottom. In our implementation, we're not using the membrane with such a big pores. We're using the pores of 0.4 micrometers, so none of the cells can go through. And as an example, we're using human PBMCs. So imagine 1-2 layers of human PBMCs on the bottom of this mesh, of this membrane, and they cannot go through. They cannot reach the bottom well. And then on the top of them, it's ideal T-cell culture medium. In the bottom, we also have the ideal T-cell culture medium. And the cells are pre-treated with calcium flux sensors. So these sensors become brightly fluorescent when calcium flux is happening. Calcium flux is an initial event in T-cell activation. It reaches the peak in just 2 minutes. And we were testing, what if we add the peptide in the bottom well? How quickly this calcium flux response is generating? And we see 2 minutes is not enough because there is some time needed for diffusion. And what is good, the diffusion is happening almost simultaneously for all this mono or B layer of the cells. But it takes approximately 5 minutes to have the saturation of this response. And then we see the fluorescent cells. We can count how many cells responded, similar to ELISpot. But ELISpot requires like a couple days. This requires just 10 minutes. So in 10 minutes, we do the fluorescent microscopy scanning of this bottom of the membrane, of this cell layer. So we basically figured that one cycle can take just 10 minutes. And I asked myself, like, for how long we can continue the cycling? And at least for 1 day. And this is how we came to our name, 100x Bio, originally 100x Bioassay, because in just 20 hours, we can run 110-minute cycles. Again, these are both approximations. We don't know, we did not optimize this method. We did not run 100 cycles. But it's our like fair estimate. And what we did, in fact, was 5 cycles, up to 5 cycles. And here I'm showing just 2 of them. So imagine, as a background, there was like couple bright fluorescent cells just activated somehow by default. But then I added the mixture of flu peptides, flu, influenza virus, and these peptides were overlapping peptides covering HA, against which most of people are immunized. So we have very high T-cell response to this mixture of peptides. And this mixture contained more than 100 peptides. So we can see in just one field of view, all the dark areas are just many different other cells of human PBMCs. So they include non-activated T cells, but activated T cells became brightly fluorescent. And here, I don't want to lie to you. It is not exactly how the fluorescent looks like. We outlined with the software, free counting software, the actual cells. So we outline and count them using the software. So we see 62 cells got activated. And it was in less than 10 minutes in this one field of view. And it's very normal to expect that if we use more than 100 peptides to some of the peptides, these cells just didn't respond. And it's normal that epitope distribution is very uneven. And then I removed from the bottom well this flu peptides, and I added 100, more than 100 of COVID-related peptides, covering the S1 domain of the spike. So we again expect a lot of T cell activation because these PBMCs were collected almost everyone in America got vaccinated. And indeed, we see 183 activated cells in the same field of view. So the previously activated ones, they stay fluorescent, but now we have extra 121 cells activated. So the idea is very simple. If we run those peptides, instead of using this huge pool one by one, we would see those cells becoming fluorescent, like slowly, 0, 0, 5 new cells activated, 10 new cells activated at different cycles. And in 100 tests, we would derive 100 times more of the data. So this is activation step, but then how we figure the phenotype of the cells and how we figure that what is the TCR sequence. So this is our ideal vision for the downstream assay. So imagine we have only 5 cells which responded at different cycles of treatment. They are rare. It is normal, and it's normal for autoimmunity. It's normal for virology research as well. So very few cells activated. Let's say it's just 5. And they activated the different cycles. But because we don't shake this plate, we even keep it stable inside the microscope. And we create the best environment around, and we're just exchanging the medium. In the bottom well, the cells don't lose the relative location. So their coordinates encode at which cycles they got activated. And what if we fix all of them? We can use different metrics, or we can use different ways of fixing them, and then run special transcriptome research. And we can see the overlap between their coordinates encoding the number of cycles which activated them and with their full transcriptome data. Again, it's not necessary to run full transcriptome. Maybe we want to run TCR sequencing plus some sort of probes against select targets. That can be 100 targets, it can be 1,000 targets, but we can very well describe the phenotype of the cells and TCR sequence. And we can give these cells some time to develop the cytokine responses, so we can look at the expression level at different cytokines, not 1, not 2, not 5. We can look at whatever these cells start to produce. And if we give these cells more time, not a few hours, but we give them, let's say, several days, 5, 7, 12 days to start proliferating, then we can help them to organize the clones. And in these clones, our sequencing data will be more reliable. And so this is our development cycle. So we started from manual assay up to 5 cycles. Now we have the robot which will be submitting the new treatment to the well, controlling the pump, and removing the previous treatment from the well, also controlling the pump. We have that fluorescent microscope, which have this scanning capability. Of course, we need to scan the whole bottom of this membrane to have the best statistical output. And then our next stage is just letting a few cells activate through, I don't know how many cycles, maybe 20 cycles, maybe 50 cycles. But then to give them some time to proliferate and run the bulk RNA transcriptome and check the TCR sequences of proliferating cells, if it works out. But in the middle, we just want to show that manual 20 cycles or so work. And downstream, of course, this is a dream idea of developing this special transcriptome overlapping with multiple T cells activated, and let's say T cell clones activated. Let me see. How we compare to the usual methods. Usual methods for T cell research, I just list the most popular ones now. They are ELISpot or fluorospot, flow cytometry with intracellular staining, or sometimes AIM, and the multiplex cytokines. So there are a bunch of companies producing equipment for all that. Producing equipment, producing antibodies, producing ELISpot kits. And there is some cost of these methods, but there is also big cost created by intensive labor. Everyone who does the T cell research, we know that it takes hours and days And also another source of cost is just access to the sample. Sample is limited and expensive. And then there are genomic methods to analyze, like focused only on TCR without real T cell activation. They're just looking at TCRs, and they already become expensive, and they already start to take a long time. Those projects take weeks, not days. And then there are complex methods, very complex ones, which combine real T cell activation with TCR data. And these, very often, they stay appropriate, how to say that? They stay inside their like company to generate the new therapies, like in a company called T-Scan or in a company called Repertoire, or sometimes they become super complex, super expensive. And because they are very expensive, they are very hard to scale. And we're trying to address all of that, mostly by multiplexing the activation. We can reduce the cost, and making our method automated, we also reduce the cost and make it more simple.
Quick question, Sergei.
Yes.
How many cells respond to multiple antigens? This was probably from the previous 2 slides back.
I want to understand the question better.
Felix, you want to unmute?
Oh, yeah. I mean, in your graph, in your table, or in your figure, it's like this cell responded on cycle 8 and this cell responded on cycle 23, but I don't really know immunology very well, but I mean, I guess multiple cells will respond to multiple antigens. No?
First of all, here we are describing the situation where we screen a library of individual analytes. So each analyte is one single peptide. So one single peptide related to some antigen, let's say COVID spike. And of course, we can expect multiple cells activated at cycle 60. We can expect 5, 10 cells, I don't know, 20 cells, whatever. This is presented for simplicity's sake, only one cell responding at one cycle. Yes.
But I just want to make sure, like the cell that responded at 60, it might also respond at 57, and you keep that information? Yeah, of course.
Here we are addressing the question of cross specificity. Of course, cross specificity can be an issue, and it is solved by different ways. One of the ways is to predict in silico where the cross specificity is to expect, and run these peptides in different wells. Second is we can, in different wells, use different sequence. In one of the wells, we add number 57 first and number 60 second. In the other well, we add number 60 first and number 57 second. So we can see, like, at least by the statistics, are there some cycles for which this change of sequence, gives us idea of cross specificity. And I think we can distribute the same sample in more wells and try to avoid this cross specificity by this way. And also, no one prohibits us from validating our findings. So we can validate whatever we found with individual peptides later on. We can validate using the standard methods like ELISpot, flow cytometry, tetramer staining, whatever we prefer.
Okay, thanks.
We have another question. How about cell death following T cell activation from Luteton?
So after the T cell activation, T cell can actually, if it's CD8, a T cell can kill the antigen presenting cell. This is what I worked with, very interesting. But T cells after activation, maybe it's a lack of my knowledge, but I think after activation, T cells should not die. They should they should activate and start proliferating.
I can take this question. I think the whole setup of our experiments is not designed for maximum activation of T cells. It's more natural since it's a mixture of T cells and antigen presenting cells, and not a continuous stimulation with strong antigen with a combination with, for example, anti-CD3, CD28 antibodies. And we know that a lot of CAR T cell companies and TCR transgenic T cell companies, they work with overstimulated T cells. They expand them in stimulating conditions, and these products are still functional, still after overstimulation, my opinion, induce activation-induced death, and then induce only few clones to survive. And comparing these two conditions, I think this experiment setup, it's a continuous stimulation, but every cycle is quite short. So there could be a case that some of the cells undergo activation-induced death. And I think the best answer would be that we need to run additional experiments to estimate the percentage.
But what antigen presenting cell are you using?
In the first experiments, we're just using human PBMCs. So we're not using the T cell antigen presenting cell co-cultures. We went the simpler way. So of course, in this cell, in PBMCs, all of the cells express MHC type 1 or HLA type 1, and B cells and dendritic cells and monocytes, they have
at some point, if you pulse the presenting cells with X number of peptides, you will be at saturation, because most of your binding in that 5-minute window or 10-minute window, is displacement of low affinity peptide at the cell surface. So you're favoring high affinity peptides, and then you're going to get to saturation. You're not going to be able to displace peptides. I'm not even talking 100 cycles, even 10 cycles. You're going to be at saturation, and your signal is going to drop. It has to drop. Otherwise, there's something else going on.
Yes, we had this question before, and we have an answer to it. So first of all, we know that by default, antigen-presenting cells always have some proportion of their MHC molecules open to be loaded. And the range is between 20 to 50% of MHC molecules are constantly open to be loaded. Second is there is also a turnaround of MHC molecules. They get inside the endosomes, get unloaded, get loaded again. So there is a recycling for MHC molecules.
Yeah, but that takes a lot of time. That has been measured, and you're going to favor binding of high affinity peptides. So the efficiency of exchange is going to drop dramatically after 5 cycles. No question.
Let me continue. So this is first 2 factors. And the third factor is we can predict which peptides have high affinity, which peptides have lower affinity. And all these 100 cycles, we can organize in a way that we're going from lower affinity peptides to higher affinity peptides. So even in the case that some peptide is getting stuck, there will be higher affinity peptide coming in the next cycle, which so let's say chemical equilibrium will be when the new peptide is taking this place inside the MHC molecules. And again,
but we have limited information on peptide binding on affinities, especially if we move to human system, where we're going to be, facing the heterogeneity of HLA on presentation. So if you want to do discovery using your system, that cannot be the factor that you input. It is just the opposite. Otherwise, you're not going to discover low affinity peptide binding on stimulating T cells. So sure, if you're working on transgenic T cells, you can bias the system. But if you're discovering, especially in autoimmunity, that's going to be very, very difficult.
I agree with you that if we have a lot of high affinity peptides in the system, they can create us some problem. However, these are 3 arguments that support our idea that this cycling is possible. First of all, a lot of MHCs are always open. Then there is a turnaround for MHCs when they get unloaded and loaded again. And third, we will try to add the high affinity peptides in the very end of the cycles. And the other thing is, yes, we still can aliquot samples. If we know that there will be high proportion of high affinity peptides, let's distribute the sample not in one well, but let's distribute in 3 and in 6 wells, and have these high affinity peptides somehow evenly distributed between different wells and used in the very last cycles.
Okay. From Peter Lindsley, do you have a sense of frequency of cross specificity?
Oh, was it a question to me?
Yes, it is.
Could you repeat it?
It's just in your chat. It's from Peter Lindsley. Do you have a sense of the frequency of cross specificity, Sergei?
We have a sense, only a sense. I'm not a huge expert in this, but from what I know, the cross specificity is a small problem for us. It's a small, like at a like high level scale. It's a very small problem, and people historically use large peptide pools, very large peptide pools. If the cross specificity would be such a big issue, no one would ever use the pool of 300 peptides. It's just we not here to propose some ideal method. We are here to propose something that's better than what's done before, and what's done before. No one ever thought of cross specificity being an issue using peptide pools of 100, 200, 300.
Sophia, do you want to weigh in on any of these questions that were asked? Or do you want to?
I added my point. That's
okay, great. And then, so if we want, in the interest of time, do you want to move forward?
Yes.
Sorry.
So I just wanted in the end to say thank you to all help that we got online and via our personal connections. So first of all, if anyone wants to start a startup company, we really got helped by taking an online course of Y Combinator, which helped us to understand how to build a venture capital-based startup. Then we really were lucky to find great IP and corporate lawyers who almost do for free a lot of company setting things for us. And if anyone starts a company, I can try to help with connections. And also, it was very helpful to find accelerator with shared lab space, because renting a whole lab would be impossible for us. But we're renting very small, tiny space where we put our own 2 pieces of equipment, but we can use all of the other equipment. And of course, it wouldn't be possible without friends around investors who believed in us. And now, while we're talking to a bigger academic community, is we open to first collaborations. And of course, for our first people who help us build a case, how this technology is applied to their samples, to their research projects, we will try to offer the very best terms. Because this technology, as you can see, it's in early emergence.
Yeah, I think that's great. And you guys are right now, well, you were in Tel Aviv and Boston, and now you're just in Boston. Is that correct?
I'm in Boston, and Sofia is now Europe-based. She's working in different cities, and we have a plan that she comes here to work on site as well.
Great. You know, I know that we had talked about the fact, you know, Sophia had shared the potential, sort of a letter to a couple of scientists, sort of, you know, laying out what might be available in terms of a potential collaboration. Do you want to do a little high-level discussion of that quickly before the very end?
Yes, high-level discussion is, for example, you have the samples that were already pre-tested for T-cell response against different peptides, different epitopes. And you have the leftovers of these frozen samples, and you want to compare your data with what our tool is generating. And if you satisfied with the quality of the data, because this can be also used as a pre-screening tool. You pre-screen using this, you screen 100 peptides, and then you can go down to 10 and validate with ELISpot. or with any traditional method. So if you want to run some small pilot with, let's say, 5-10 frozen vials, we open to discussing the exact terms. But it's not going to be something like crazy expensive. Maybe it's going to be even free. It depends on how much money we will keep raising.
Yeah, I think it provides a really nice opportunity to sort of test, you know, as you said, sort of your extra frozen samples or some other, you know, from previous experiments, sort of test this new tool out. And I would encourage people to sort of reach out for one-on-one discussions to learn more about the possibilities of these two young entrepreneurs and their idea of kind of making this space a little more easy to use and less expensive to use, and perhaps you know, there's a place there for intersection with type 1 diabetes samples. So in the interest of time, I'll let everyone reach out directly, contact at 100xbio.com for more information. And thank you again for your presentation today, both Sergei and also Sophia. Appreciate it. Best
Thank
of
you
luck
everyone
to you.
for your questions, and thank you, Monica, for this opportunity.
Absolutely. Have a great rest of your day. Bye-bye.
Have a great day. Bye-bye.